Glycolysis, most commonly known to the community as Miss Sugar Sever, is sitting in her rocking chair in her humble abode, the cytosol. She holds 2 knitting needles and seems to be deep in focus. The way she knits is peculiar due to the fact she's not merely knitting a sweater, but rather doing her very, very, very important job, which she refuses to retire from for a plethora of reasons. One of those reasons being her love for her occupation. Why don't we ask Miss Sugar Sever to explain exactly what she's doing, for I highly doubt anybody could do it better.
Oh my! I didn't hear you come inside. Take the moment. I'm completing an order for my employee at CAC. What's that? You'd like to know exactly what I'm doing? Well, take a seat. Now, you don't look like you're from around here, so it might seem a little strange as to why I'm ripping apart this glucose sweater. You see, this glucose sweater is made up of 6 carbon atoms. We need to hack through it using 10 trusty enzymes to get to the 2 pyruvate molecules, which can be used to make energy. In order to do this, we need to invest 2 ATP or 2 adenosine triphosphate into our glucose sweater, converting them into 2 ADP, and destabilize the glucose, making it unstable and ready to split. But before the split can happen, this 6-carbon sugar must be rearranged in order for its enzymes to be able to split it properly. Once split, it becomes 2 3-carbon molecules called pyruvate. Now I just need to rearrange and oxidize these, which, as you can see, releases hydrogen atoms and high energy electrons. Oop, right on time. Strangers, this is NAD+, carrier molecule. He's here to pick up the hydrogen atoms. This will form 2 NADH.
I'll bring these to the transport chain.
And at the same time, I will make 4 ATP, substrate level phosphorylation. But because we spent 2 in the beginning to start the whole process, we have a net gain of 2 ATP. A transport protein will be by to pick up the pyruvate for pyruvate oxidization.
This transport protein is traveling to the mitochondrial matrix. Once here, an enzyme converts each pyruvate molecule in the box into acetic acid, a 2-carbon molecule. Another NAD+ picks up electrons and hydrogen atoms to become NADH, heading off to the ETC. During this process, 1 carbon is released as CO2. Then coenzyme A picks up acetic acid to form acetyl-CoA, which can now be delivered to the CAC to be processed.
The acetyl-CoA is dropped off here at the CAC, and when it gets here, it bonds with oxaloacetate, which has 4 carbons. Together, they form citric acid, which has 6 carbon molecules. This starts up the cycle. Citric acid then undergoes a series of transformations, losing hydrogen and carbon dioxide atoms along the way. Hydrogen ions and high-energy electrons are transferred to carriers like our old friend NAD+, and others like FAD, and turn them into NADH or FADH2 before delivering them to the electron transport chain, where they generate the bulk of ATP formed from this process. This process will result in 1 ATP, 3 NADH, and 1 FADH2.
We electrons are passed from NADH to oxygen, moving protons from the matrix to the inner membrane space, so are dropped off by the FADH2. We use our energy to move hydrogen ions into the inner membrane space.
See, me personally, I think we have the most important job of the cellular respiration process. I mean, the electron transport chain is where most ATPs are made, and it's called electron transport chain. Like, hello, talk about star player.
She said that? That's adorable. Hold your breath right now. Are you feeling comfortable? Yeah, didn't think so. I'm the final electron acceptor in the electron transport chain. My role is crucial for generating ATP. It accepts low energy electrons and protons to form water, allowing the chain to keep running and maintain the proton gradient that powers ATP synthesis. Without me, the electron transport chain stalls, preventing efficient ATP production and leading to cell death.
Yeah, those girls argue a lot. Maybe once every workday, which put in human terminology is 1,000,000 times every second. That is a lot to fighting. Would I consider myself a key player? Yeah, no. We H plus molecules get carried around a lot. We did act to go with the flow. Follow the laws of thermodynamics and, high concentration to low concentrations. We don't love crowded areas. So a lot of us up here, we all have to leave through the revolving door. Back down to low concentration. I mean, maybe I'm a key player. All of the spinning of the door produces ATP, and that wouldn't have happened if I didn't want to go home. We make up to 34 ATP in this process, so maybe we are the star players.